everybody see, or would you like some darkness on the slide? Okay, let's begin like that and see what happens. Okay, so Boker Tov, good morning. My name is Chaya Kalheim. I'm from the medical school, the Department of Medical Neurobiology, and I'm a member of LSEC, and I usually give you only 4 hours of a course, which, as I said to some of you outside, is a little bit frustrating, but at least, this is what we have, and let's enjoy it. So it's going to be a little bit of an introduction, and you should bear with me, and please do ask questions. Let us do it as a kind of conversation more than as a frontal lecture. So basically the idea of giving you a little bit of developmental neurobiology is for you who are physicists and mathematicians and computer people and people who maybe are aimed to study higher brain functions. Our idea in development in general, and in this sense neurobiology is only one aspect of general development. The idea is trying to understand the generation of complexity, because every system in the body, and our body is extremely well patterned. We have bilateral symmetry, we have a head here, and this is not by mistake. This is an evolutionarily important issue which we may tackle next Thursday if we have time. So the composition of our body, the form of our body, and all of this is really evolutionarily regulated and controlled, and nothing happens by chance. Nothing happens by chance, everything is tightly regulated. The nervous system is only part of this regulation. It does not develop in vacuum. It develops based on many other primitive systems which the body contains, which are called endoderm and mesoderm, namely the progenitors, those cells which are going to generate our gut and our liver and our lungs and our muscles and our cartilage and vertebrae. So the nervous system is not the first system which develops in the body, by no means. And it is dictated, its formation, the way it is generated is, in a sense, dictated by the other tissues. So being developmental biologist, or even being developmental neurobiologist, means understanding the interaction and the crosstalk, which is very intimate, between progenitors of the nervous system. The word progenitors is familiar to you. The progenitor is a cell which is going to generate a neuron, a glial cell, and so on and so forth, or any other type of cell. Progenitor is the primordial cell from which an adult derivative is going to form. So progenitors of the nervous system cross-interact with additional progenitors in order to get differentiated, in order to migrate, in order to get patterns in the proper way. So this is something we are not going to touch upon, and for me it's the most important thing, because we in the lab are studying currently, and in my own career I began studying differentiation of the peripheral nervous system, and we go earlier and earlier and earlier on in development as I age personally, because I find that these are the most challenging problems for us as developmental biologists. So I have written on the board, I don't know if you can all see, but I have written in the board a series of different processes which I termed induction, neural induction, fate determination, namely what do I become when I am grown up, cell proliferation, the fact that cells undergo mitosis, they divide in order to generate the final size of the body and of the spinal cord, peripheral nervous system, or CNS, cell migrations, the formation of layers, which, for example, characterize the cortex, and so on, axonal growth and guidance, neurogenesis, gliogenesis, apoptosis, cell death, which is a very important process in nervous system development, synaptogenesis, and finally the formation of circuits, which will enable all kinds of behaviors, from the primitive ones till the most sophisticated ones. So I'm going to touch upon all of those in a very superficial manner, and as I said, you should bear with me on that. So the first slide I like to show everybody always in the course is this slide, which is a general slide, not related to the nervous system in particular, and I would like your help to try and understand it. So for those who do not understand Hebrew, this is a fish, this is a salamander, this is a turtle, this is a chick embryo, this is a rabbit, and this is a human being. So what do we see here? Can you help me out? As I drink tea. What?
3 different stages in embryonal development of each of these different animals?
That's right. And what is peculiar here for each one of those stages or each one of those species? Yes?
Tail.
The tail.
First, yeah.
Okay.
Second.
Okay, so let's keep in mind the tail. What else?
They all begin the same.
They all begin the same. I wouldn't say the same, but they all are very similar at the earliest stage of development. And then, which is one of the important principles, look at this early stage of development. It's not that early, by the way. If I may just give you a glance of aging, this in human development would correspond to 1.5 months after fertilization. It's not that early. As a matter of fact, in I don't know about the rabbit, but in chick development, this would be like 2.5 days of development, okay, out of 21, and so on and so forth. So at early development, or relatively early development, embryos look the same. Embryos have a tail, that's right. We also have a tail. Not only that embryos do have tails, what else do human embryos have? And this is already a hint that also fish embryos have. I gave you the hint by saying fish embryos.
Gills.
What? Gills, exactly. Where is our fish? This is the fish, and you can see here 1, 2, 3, 4, 5 pairs of gills because they are symmetrically localized on both sides. We are just looking at the side perspective, so we see only one, and we also have gills. The big difference is that the gills in the fish are going to be and are the main respiratory organs, whereas in our case, gills do not have any respiratory function, and they will transform during development into different derivatives, such as part of your thyroid, such as the incus, malleus, and stapes in your ear, okay, all these little bones in the ear, and all kinds of endocrine derivatives, which are, I mean, really cute and very, very, very complicated. So the important point here to remember is that during early development, most embryonic types, most embryonic species resemble each other, not only physically, not only in terms of their form, but also in terms of their size. If you go to an elephant embryo, it is going to be very similar in size as well. And it is only at later stages that it is going, obviously, to diverge in terms of growth. And then the divergence begins, and what do we see here? They are very different, and let me just go a little bit faster. The thing that strikes us the most, at least to me, is the issue of cephalization. Okay, one of the most important things that happened in evolution, and which reached its peak in human beings, is of course the development of the brain, which, as you can see in a fish embryo and even in lower animals, lower vertebrates, there is a kind of brain, or even in lower vertebrates, there are like ganglia, which are localized segmentally along the body axis, but there is no central processing of all the information. Whereas during evolution, we have grown a brain which is increasing in complexity. And obviously, in the functions it may exert. And this is called the principle of, let me remember, I'll remember, it's not, I'll remember in the break. Okay, never mind, it's a German scientist who said phylogenesis or ontogeny, namely the development of an individual, recapitulates the development of the evolutionary tree. So this is an idea which is only partially correct, but never mind. So this is something which is important, but at the level of the individual, there are several processes which are common to all the tissues that the embryo is concerned with, and this is cell specification, the proliferation, epithelial to mesenchymal transitions, namely the fact that different cells are arranged like little soldiers, one attached to the other, and at a certain point they acquire a motile capability. They become mesenchymal. If I may draw this on the board, cells are born as an epithelium. This is a kind of structure which medical students learn in histology courses, and these cells will then become mesenchymal, which are freely motile cells that are able to migrate. Because many of the cells in our body, including those of the nervous system, end up in places which are very distant to their source, to the place where they were born. So this type of process, EMT, epithelial to mesenchymal transition, and then the reverse, are a very important and crucial part of morphogenesis, of the formation of the embryonic form and the adult form, of course. This is followed by migrations, by fusions between different tissues, by budding. Think about a glove, okay? As you push your fingers inside, there is like a budding from an initial tube. This is the way many things in the nervous system, and mainly, for example, in the gut, are formed. How is our liver formed, our intestines, our lungs, by budding, then rotations of organs, which will push those to become in different places, and apoptosis, or programmed cell death, upon which we will touch during the second hour. So there are a lot of very important processes that we have to tackle if we wish to really, really, really understand how the body is shaped and how it develops. But let's go to the nervous system, which is our favorite one. And the nervous system is basically formed from a layer of cells which is called the ectoderm. The embryo, let's say the human embryo, by the 2.5 weeks of development after fertilization, when the potential mother barely knows that she's pregnant, the embryo is already formed of 3 layers. And I would like you to think of 3 coins, okay, one on top of the other. The upper coin, okay, so it's something which is in 3D, not in the plane of a board. The upper coin is called the ectoderm, the middle coin would be called the mesoderm, and the lower coin would be called the endoderm. So the ectoderm is the origin of the entire nervous system, and also the skin. All our skin and skin appendages, okay, like hair or feathers or stuff like that, are all coming from the ectoderm. The mesoderm will generate muscles, vertebra, blood vessels, and so on and so forth. And the endoderm will generate your gut and your respiratory system. Okay, the genitourinary system will be generated by mesoderm. So for us, the ectoderm is the important one. And the first step in the development of the brain and the spinal cord and the peripheral nervous system is the following. So what you're looking at here is a scanning electron micrograph. Let me turn down the light for a sec. Okay, so that you can appreciate this better. Is it okay? So what you can see here are different stages in a process which is called neural induction. Namely, how the ectoderm, which is fated to become either epidermis or neural tissue, how does it become neural tissue? Of course, we are going to skip an enormous amount of molecular material here, but let me just give you the idea of it. So this would be here the ectoderm, and this is like looking at the embryo from its backside. The ectoderm is a 1-cell thick layer of cells, okay, and we'll touch upon that in a few minutes. And what we see is that upon a certain induction of a tissue, which is called the notochord, can you see here a kind of circle? You see it better here. Do you see this? This structure is like a road, okay? It's like a cord which spans the entire length of the embryo. It is called the notochord. The notochord. In different embryos it is called a little bit differently. And the notochord is mesoderm. It's not ectoderm. It's mesoderm, and this brings me back to interactions between the different tissues. And this magic structure, which is called the notochord, induces the formation of neural tissue from the ectoderm. Namely, a substance coming from here, which is called sonic hedgehog, and we'll see it in a few minutes, induces somehow the formation of a neural tissue from the original ectoderm, and this will result in the thickening of this tissue here when compared to what is going to become the ectoderm afterwards. So this is the ectoderm, this is the mesoderm, and this would be the endoderm, and this is the notochord here. So the first thing is the thickening of the ectoderm that is now called neuroectoderm. And the neuroectoderm cannot become skin anymore because it is already neuroectoderm. And this is to illustrate how dynamic are the processes in development. So from interaction between A and B, we get C. And C is not A and not B anymore. So this is the whole idea. How do you create a new structure from the interaction of two pre-existing tissues? Okay, and this goes over and over again. This is a leitmotif in development. So the second reflection of the induction of the neural tissue is not only its thickening, but because of reasons I'm not going to develop now, the tissue is going to undergo a kind of grooving, okay, a kind of invagination, which you can see also here as a different level of the axis. Okay, so this is ectoderm, this is neuroectoderm, and this is ectoderm again, and finally this neuroectoderm is going to fold upon itself, and the dorsal edges of this neuroectoderm are going to fuse right with left and generate a tube. This is the way your entire central nervous system is formed. Like a tube, think of a baguette, okay, which is a long tube. So the tube is on the entire length of the body. So in the body, in the trunk and lumbosacral regions, it's going to generate obviously the spinal cord, and then by different proliferation issues and differential growth processes in the rostral portion of your body, is going to create a brain because it's going to expand much more than at the levels of the spinal cord. And this structure, as we are going to see later on, is also going to generate the entire peripheral nervous system. How? We'll see later on. Okay? So this is called now the neural tube. By the way, neural induction is much more complicated than the interaction between notochord and ectoderm. I just gave you a hint of that. So you should know that what I'm telling you is not lies. It's the truth, but it is only a partial truth. Okay? So this is what we call in Hebrew a sheker lavan. So you have to bear with me because otherwise, neural induction is a subject of a course. Okay, we know which are the neural inducers. We know how it happens in different types of embryos. It is a very elegant process which, people dealt with for tens of years, and it was just in the latest decade that we really came to understand how this happens, and may I say not completely. So what we see here is exactly the same process. Okay, we see the ectoderm. We see it, for example, this is the neural ectoderm called neural plate, as long as it is a plate. Okay, a sheet, and then we see this neural groove stage, finally the neural tube stage, and this, which is going to be the skin, once the tube forms, the skin or the ectoderm is going to, get isolated, regenerate on top of it, and it will really form the skin on top of your spinal cord or brain. And the next point here is how to transform this tube, which is a naive tube, into a tube which has different functions along its dorsoventral extent, as well as different functions along its longitudinal extent. So let us deal with the cross-section. Let us deal with the ventral to dorsal extent, because we know that the dorsal portion of the neural tube is going to be the sensory portion of the central nervous system, which will receive and integrate sensory inputs coming from your skin, your muscles, coming from proprioceptive stimuli, etc. Whereas the ventral portion is going to be the motor portion, which is going to be formed by interneurons and by motor neurons that are going to give our muscles different orders: move, don't move, do this and do that. And then, of course, you have a lot of interneuronal type of cells which relay the sensory information towards the motor information, and all of this has to be properly formed in time and in space. How does this get formed? This is a very difficult thing. I'm going to try to give you a few ideas. So the first thing you have to know is, again, that there are interactions between the notochord and the ventral tube, the ectoderm, and the dorsal neural tube. And in fact, these are going to dictate the dorsoventral patterning of the neural tube, which is going to be formed first by a structure which is called the floor plate, a kind of glial-like structure, and also by a region which is called the roof plate. And then this is going to be subdivided into these color-coded regions, which are formed by different neuronal subtypes. How does this occur? So again, we are going to go back to this molecule which is called sonic hedgehog, which is produced and secreted by the notochord. So let me just show you a very nice experiment that has been done in chick embryos. In chick embryos on which we work in the lab, we can do a lot of molecular things, but we can also do something which is not possible to do in mouse, and this is, or in higher vertebrates, which is microsurgery in the egg. So we can change tissues. We can change genes in different tissues and then graft in ovo within the egg, and then cover the egg with sellotape and put it back in the incubator and really hatch it, or keep it until different stages and then fix it and analyze the outcome. So many things, very, very elegant ones, can be done in this type of embryo. And what we see here is the floor plate, and this is the notochord, okay? So we know that the notochord induces the floor plate. The notochord is the first source of sonic hedgehog, and once it induces this area called the floor plate begins as well synthesizing sonic hedgehog. So we are going to have two organizing centers. The first is the notochord, and then both notochord and floor plate. So what happens if we graft ectopically? Ectopically means in a place which is not the normal one. What happens if we graft an ectopic notochord and tab in the dorsal portion of the tube, where there is normally not any notochord? Can you tell me? What has happened here? What did we do?
Another floor plate
Right.
for them, induced for them.
Right, exactly. What we have done here is we have induced an ectopic floor plate. And this is a demonstration that basically the notochord has an inducive or an inductive ability to create ectopically in a region in which it is normally not produced another floor plate. Okay? So what happens if we do this exercise? We put two adjacent notochords. Okay? We do exactly the same. We induce two ectopic floor plates. So in fact, instead of the floor plate being here, we induce two floor plates adjacent or dorsal to the two notochords that we have formed. So, right, yes.
Question
Yeah.
about the resource method. Can you study it only on tissues, or do you have a model of the different molecules in the computer?
Let me come to it now. Okay, yes, the answer is yes, and more are developmental neurobiologists using modeling to try not only and mimic what happens, but obviously also to predict how different molecular networks would operate. And we are also very much interested in that, but we do not study the ventral part of the tube, we study the dorsal part, on which we are going to talk on Thursday a little bit more. Yes, absolutely. And this is becoming more and more fashionable. Many people doing it at a very high level, other people doing it quite in a mediocre way, as usual. So let us try and look now at motoneurons. Motoneurons are those which have an output towards your muscles, for example. So these are, forget about this one. These are sensory ganglia, which are also stained by this motoneuron marker whose name I'm not going to give you. Okay, so these are motoneurons, and you also can see the floor plate. So what happens is the following. It appears that the notochord at a very high level of sonic hedgehog, of this molecule, which I'm going to call in a minute morphogen and explain myself, will induce a floor plate. At a little bit lower level, it will induce these three interneurons. This is a type of neurons existing in the spinal cord, which are these ones that are unlabeled here. And at a bit lower level will induce motoneurons. This means that what we have here is a gradient of a molecule. And cells exposed to different concentrations of this determinant, of this protein, will become different cell types. This type of molecule, and sonic hedgehog is that molecule, is called a morphogen. So a morphogen is a concept. It's a molecule which dictates different fates when the cells are exposed to it at different concentrations.
Just a single molecule?
Excuse me?
Just a single molecule?
Just a single molecule. It's much more complicated than that. We'll get to it in a second. Okay? Yes, but the beauty of development is that we know about 10 morphogens. Sonic hedgehog, BMP, Wnt, FGFs, and maybe I'm missing a few. That's all. And these are enough to pattern your body. So how do they get complicated? You will see in a minute. Because the morphogen only gives you the first hint. And this is then relayed upon to additional molecules which act what we call downstream of the morphogen. We'll see that in a minute. This is how we can make a simple thing extremely complicated. And I think maybe this is clearer for you with a physical or mathematical background more than for me. So this is very easy to put into equations, I guess. So what happens, basically, okay, let me skip this. It's not important. Okay, so what happens if we put these two notochords here? What we get is an entire floor plate. So we basically have completely impaired the formation of a gradient. So instead of getting here this gradient of different molecules, we are here. If we have a lot of sonic hedgehog coming from the notochord, what we do is we basically oblige the system to make a very, very large floor plate. And this is to see sonic hedgehog. This is an antibody. No, this is an in situ hybridization, excuse me. In situ hybridization is a reaction that we perform on embryos or tissues in which we recognize the RNA encoding for a certain gene. Okay, so this is RNA encoding for sonic hedgehog. Where do we see it? In the notochord and in the floor plate, as I told you. So you can see the notochord in the entire embryo. Okay, this is the wing bat because this is a chick embryo, and this is the entire axis or part of it, and this is the notochord and the floor plate. So sonic hedgehog, in terms of its expression and its production, is very, very, very restricted. But in terms of the release of protein, because sonic hedgehog is a protein which is released and exported from the cell, it basically creates a gradient which arrives up to this area of the neural tube. So it is produced here, but it is secreted and basically migrates as a molecule thanks to cargo proteins. It has friends. Sonic has many, many friends. It's a very nice molecule to his peers. Sonic has like 20 different molecules which enable it to get secreted. Other molecules, the names are obviously not important in this course, enable this molecule to get expressed on the cell membrane, enable this molecule to get proteolytically cleaved in different areas in order to be secreted. Additional enzymes which add different moieties to the molecule, like palmitoic acid or cholesterol, namely fatty acids, which are very important for its activity. Additional molecules which upregulate the activity, other molecules which negatively regulate its activity. So this is how you take one morphogen and we basically can integrate it into different types of activities and different concentration gradients at will during development. Okay.
What's the developmental source of this morphogen?
The developmental source, it is produced in the notochord and in the floor plate. What do you mean by developmental source?
Like,
Other than that? this is a teleological question. It's like why is this neuron, I mean, giving me an order to move my muscle? We do not know how to deal with teleological questions. Why it is produced there? It is produced there. Science doesn't deal with why. Science does deal
No, but
with
you
how.
can ask the same developmental questions.
Let me advance a little bit, okay? The question of why is sonic hedgehog produced in the notochord and floor plate? Sonic hedgehog is produced in the notochord and floor plate. When you deal with development, you are not an engineer. You are not somebody who builds a system. The system has been built and optimized for you by evolution. What you are basically, as a biologist, you are a Sherlock Holmes. You try to uncover the secret which exists. It's very difficult to ask why. I think it's not even a legitimate question in development or in biology. It's the how that we are interested in. This is how it works, and we want to understand how does it work. Why is teleological,
Yes,
sometimes
I'm asking how does this
philosophical.
specific molecule get introduced and not some other molecules?
But this is why it is produced there. It is produced there. I don't think I have an answer for you. Why it is produced there, I don't know. If it weren't sonic, if it were wind, would you ask me why wind and not another molecule? Because it's wind.
I guess the question is like, what makes the notochord, there's probably some sequence of events that gets the sonic hedgehog
Not that we
the
know.
notochord.
Not that we know. Sonic hedgehog is an evolutionary, very ancient molecule. It is already produced in the fruit fly, in Drosophila melanogaster, where it also patterns, for example, the wing and parts of the nervous system. So it's an extremely, extremely ancient molecule which underwent an evolution to create patterns. Okay, so this may be the closest answer to your question. More than that, I can't give you. Why this molecule and not the other? We can ask that for every single tissue in the body, in which specific molecules and not others are there. Why? We know that sonic in the human being and in other animals is also patterning the limb. Okay? You know that your limb has a pattern. This is distal, this is proximal, this is dorsal with your thumb, this is ventral. Sonic is involved in patterning your limb. And sonic is not the only important one, because against sonic there is BMP and Wnt here, and they are in a war all the time. BMP says dorsal, sonic hedgehog says ventral, and they keep fighting all the time until the nervous system is patterned. Okay, so why BMP in the dorsal? Very difficult question. So let us see how sonic works, and this is the way every morphogen works. Okay, so we have here 3 cells. These cells have receptors, for example, for sonic hedgehog or for any other morphogen. And these cells have also different outputs, a secondary line and a third line. And these are different cells that are going, or different states that are going to get developed. So the first thing we see is that there is a signaling molecule which is created in a gradient. The closer you are from the source, the more you are going to get, right? This is a logical assumption. So this is high and this is low. 3 molecules here, 2 and 1. So the next step is that these molecules are going to bind to its receptors. There are 2 receptors for sonic hedgehog, maybe even more. One is a positive receptor, one is a negative receptor. There is feedback among them, very complicated thing. So here you see that at the high concentration, you have highlighted 3 receptors. At the lower concentration, there is only a partial occupancy of the receptors. So these receptors begin exerting a signal which is going to be relayed upon a series of primary transcription factors. These are proteins which enter the nucleus, bind to different DNA sequences, and are able to exert the transcription of additional type of molecules. So look at the hierarchy. There is a hierarchy here. Sonic doesn't work just by itself. It goes to receptors, it goes to primary transcription factors, then it goes to target genes, those which are produced upon entrance of sonic into the nucleus, and as you see, the target genes that are produced by high occupancy are different from the target genes that are produced by intermediate and low occupancy. And after that, there are secondary transcription factors produced by the cells, which are basically going to dictate different phases. So what you see here is that Sonic gives the initial order. Guys, this is it. And then, based on the concentration of Sonic, there is a whole hierarchy of activities, which are molecular activities, which are differential at different concentrations that are going to dictate different phases. So this cell is going to be different from this one and from that one. Let's see what happens in true life. Of course, this is going to be translated, as I said, into different cell identities. Let us look at it this way. There is a gradient. Okay, sonic hedgehog is higher here and lower as we go far apart. Then sonic hedgehog here is going to create in a graded fashion the synthesis of molecule blue, pink, and yellow. Then it is going at the same time, as I said, because it fights with BMP in the dorsal tube, to inhibit the synthesis of molecules red, violet, and green, which are basically produced here. So you have a couple of molecules which are going to be synthesized here, and a couple of molecules which BMP and Wind will induce in the dorsal part. So it is going to be positive towards the ventral ones, but negative towards the dorsal ones. So what happens here? This is going, basically, if we look at this area, to create at a certain level of the tube, to create a fuzzy type of border between the red molecules and the blue molecules, namely the ventral molecules and the other ones. What is going to happen now? We have to sharpen these borders, because every cell type is going to express a certain repertoire of molecules and not another one. So this is not a matter of sonic anymore. These molecules here, gene red with gene blue, are going to interact in a negative fashion with each other, and then this is, in biology, this is a sign for inhibition, and this is a sign for excitation or induction, okay? So molecule red is going to say, whenever I'm here, you don't exist. Molecule blue is going to say the same. So they fight. As BMP and sonic fought earlier on, these molecules, which are the result of BMP and sonic signaling, will fight in turn. And then the output is going to be the creation of a sharp border. And this sharp border means the creation of different cell types. And let me give you now, and for example, you can see already, so the floor plate will express blue, pink, and yellow. These three interneurons will express this repertoire of genes, and the motoneurons will express this one. So look how we create different cell types. We create them first by the activity of a morphogen, second by the creation of a hierarchy of transcription factors, which then interact among themselves in an inhibitory fashion that will finally sharpen the boundary in a given territory to create different cell types. So each cell type is not going to express a single transcription factor. Each cell type is going to express a network of interacting transcription factors, which makes it different from the other. And this means that we don't need many molecules to make a cell. We just need the proper interactions between a finite, limited number of molecules. Guys, we have 25,000 genes in our body. This is very little. So how do we create such a complexity? By positive and negative interactions and by combinatorics of molecules. Namely, it's not one gene, one protein, it's one gene, many proteins by different splicing events, and it is also different interactions. So the secret for creating complexity is interactions between genes and interactions between different combinatorics of different genes. So you need very few genes, but in different dosages And another thing which is important, different times. I haven't spoken about time, but time is a crucial point in development of the nervous system. I'll tell you why. For example, if we give the notochord, let's say sonic, very early on, when the tissue is naive, we create a floor plate. But if we give sonic at a later stage, the floor plate is already refractory, doesn't respond to sonic any longer. It doesn't need sonic any longer. So the characteristics of an inductive process is that it is limited in time. And in fact, you need to give sonic or any other molecule for any other process with a kind of curve like that. And there is a maximum, a peak of responsiveness, and then responsiveness is gone. Why? Because the system is busy with the next step. So there has to be a system that shuts off responsiveness. So time and space and amount are all integrated within the cell. And what does keep us busy now in our day? We know many molecules. We know many of the downstream genes that operate, but what we don't understand yet, and this may be a nice project for mathematicians and people dealing with modeling, how does a cell integrate all these inputs in time and in space in a correct manner so that it provides us with the right output? Because I could argue that it is equally appropriate to get, for example, exposed to a lower amount of sonic over a longer time than to get exposed to a higher amount for a short time. And in fact, we know that to create a floor plate, you need to bombard the cell with sonic, but for a very short time. To create a motoneuron, you need more time, and you need a lower amount. So we know, and there are new data appearing now in the literature, based mostly, unluckily, on in vitro studies, because it's very difficult to change the form of a gradient in vivo, and so we are trying to do it, and then try to change the fate accordingly. So how the cell integrates time, space, and amount of a molecule, this is still quite a mystery. Okay? You tell me when to stop. Let me just finish this idea, and then we'll take a break. So here you can see an example of different cell types and how they are created. So for example, sonic produces class 2 transcription factors, never mind what they are, okay? These are the names: NKX2.2, 6.1, DBX2, PAX6. This is a whole field, okay, that I'm not going to bother you with. And on the other hand, it inhibits the class 1, which are the dorsal ones. So as a result of producing this type of factors, you have then the fight between class 1 and class 2. There is a cross inhibition, and then neuronal shapes are created. These are V3 motoneurons, V2, V1, V0, all these are interneurons. And look at them. These are all of the different cell types in the spinal cord, okay, that we can visualize actually with different antibodies to the different molecules. So beginning with a very naive epithelium, the neural tube, we end up with different fates along the dorsoventral extent. I'm not going to talk or to discuss the longitudinal extent, which is basically dependent on many other cues, which are partially very different. Okay, yes. I think it's a good time for questions, and we are going to make a 10-minute break and continue. Okay.
So when they do kind of the two factors or whatever factors are inhibiting each other, is it always like that one wins and the other lose, or can it get to a condition where they can't actually decide and the cell die or something like that?
No, basically in development at this stage in which fates are determined, there is very little cell death. Almost no cell death. It goes beyond 1%. Cell death is maximal in the tube at much later stages when the neurons have already been formed, when they send their axon, and when there is synapse elimination in the periphery. Namely, when there is already a crosstalk with the periphery, and that the CNS has to adapt itself to the site of the target it is going to innervate. So no cell death. There is a competition, and the factor that wins, the work there, and that sonic is not backed up by additional factors which are existing there, because receptors for sonic like touch and smoothness continue to be there, and this strengthens the idea. So each crucial event in development does not happen by one single mechanism, it happens by many mechanisms, which back up each other to ensure that it is appropriate. Because what is amazing, what is amazing is that even if you have processes which are not entirely deterministic, because this fight between transcription factor is not entirely deterministic, there are very few mistakes that are made in development. The fact is that we are quite perfect. I mean, not perfect in the sense that we're perfect. Perfect in the sense that the embryo develops, I mean, most of the embryos develop very well. And those who are not, I mean, the nervous system is not an issue for abortions, right? Usually these defects are seen later on. Very, very few defects in the formation of motor neurons and sensory neurons. Most of the defects in the nervous system are seen in closure of the nervous system, spina bifida, stuff like that. Very, very little errors made at this level.
So in this stage, all the cells in the nervous system are differentiated?
No,
or
nothing is differentiated yet here.
Okay.
This is a very good question. Nothing is differentiated. We are talking here about progenitors. We are going to talk about differentiation later on. These progenitors are all of them, cells which still proliferate, which still undergo mitosis, but we already may call some of them neuroblasts. Namely, they are what we call committed progenitors. They are not naive. Okay, the moment that they enhance a certain transcription factor at the expense of another means that this cell is already fated to become X and not Y. Okay? It still proliferates. It did not, a neuron is not a proliferative cell. A neuron is a post-mitotic cell, a cell which already withdrew, exited the cell cycle. Okay? And these are all proliferating progenitors. So what we distinguish in development is something very beautiful. There is a kind of hierarchy. Think about a triangle, and upside down, okay? The higher you are in the hierarchy, the more primitive you are. And the lower you go in the hierarchy, the more differentiated you are. So these cells are somehow in the middle.
And so stem cells. So where
would you put stem cells?
Yeah.
We have this hierarchy. Okay, guys. Where would you put? This is a developmental hierarchy. Where would you put a stem cell?
Very high.
So stem cells are before the stage that we saw here, before they got to this process, they were stem cells.
Yeah. Having said that, stem cells is a very complicated issue. I mean, I give a course on stem cells. There is the ultimative stem cell, which is the zygote, the fertilized egg, right? Why?
Because it can become any cell.
Yeah, because it becomes the entire organism. So it's the ultimative stem cell. This is what we call an embryonic stem cell. But literature uses the word stem cell also for satellite cells in the muscle of adults. Okay, so satellite cells in the muscle of adults are stem cells of the muscle, what we call tissue stem cells, which are able to regenerate in the body only into muscle. If we take them into a dish, they will also give rise to adipose tissue, human, cartilage, and stuff like that. But in the body, they will generate entirely muscle. So these are also stem cells. Be very careful with the term stem cells. So I like, for my students, I like to call only this one stem cells, and the moment they have entered already a certain program, I call them progenitors. And progenitors can be still quite naive, but they can also be very restricted. So they are very many degrees of progenitor development. So these cells we are dealing with in the spinal cord or in the tube or in the brain, whatever area you're looking at, are what I would call restricted progenitors, maybe even committed ones, that are going to differentiate along the neuronal pathway, but did not yet. Okay? So think about something interesting. For example, it's like a couple which lives together. They are not married. They are not differentiated. I mean, nobody has to know that. But they live together, they share their lives. They are committed to each other. But they don't have the famous gradient. Okay, so this is a kind of committed progenitor. And when you get married, you go to the Rabbanut or you go to Cyprus or whatever, then you are married. Okay, so then you differentiate. You put up a new right, you begin synthesizing acetylcholine or norepinephrine, you make a synapse, you are a differentiated neuron. These cells are far from differentiating here. But this is where the important decisions are made in a cell. It's not the differentiation, the important decision. The differentiation is the end step of a long journey of commitment, of deciding what am I going to do, right? Okay, so if you got this point, I think I am okay. Sarah, let us take a break. More questions? We can continue talking in the break.
לכולם נראו בערך דומים, אבל אמר שאין לוקח להם זמן שונה להגיע לשיח הפטישות, אפילו שהם כן דומים של הקהילה, כאילו לא למה כמו לאיזה מטרה, אבל מה גורם לזה
מה גורם לזה שלחיות שונות יש
מה שונה להגיע למצב דומה?
תראה, בעכבר נולדים תוך 21 יום, צ'יק גם כן. לעומת זאת, כשאתה מסתכל על צ'יק ועכבר, צ'יק מתפתח מהר יותר, הילד גדל יותר. חיות יכולות להתפתח מהר יותר, יש להם פחות קומפלקסים. לצ'יק אין קומפלקסים. אני לא דואגת להתפתחות של קומפלקסים. אצלנו דואגת 9 חודשים, אצל גיל אני יודעת שנה וחצי. זאת אומרת, יש חשיבות לגודל, כן? חיה חייבת להגיע לגודל מסוים. האם לא מתחלקים כל כך שונה בין החיות השונות. האורך הסלסייטי הוא די דומה. הוא די דומה. ועל כן, כשאתה צריך ליצור פילון שנולד, אני אומרת, עם 100 קילו, בן אדם עם 3.5 קילו, או צ'יקן, אתה חייב, על כן ייקח יותר זמן, והזמן הזה הוא לא נמדד בשלב הראשון, הוא נמדד למעשה בשלבים שאחרי ההחלטות האלה. ההחלטות האלה קורות די דומה בספישס השונים.
זו השאלה שלי, כאילו לא סוף התהליך של 9 חודשים, אלא ההתחלה כאילו אמרת שלהגיע למצב, כאילו ממש לשלב הראשון, לא ראשון, אבל התחלתי, עם הזונה, שזה לוקח פעם חודש וחצי ופעם יומיים, וזה מגיע לאותו שלב. אז איך זה להגיע לאותו שלב לוקח זמן שונה את השאלה?
בגלל שגם תהליך הפרטיליזציה, תהליך ההסלמות, הוא מאוד שונה. יש הרבה דברים שונים בהתפתחות. הביצית של התרנגולת, הביצה מתפתחת נורא שונה.
או לא השבוע, מאז תחילת
אנחנו חיות שהביציות. העמדה היא הביצה, זה שונה לגמרי. יש תהליכים ראשונים שלוקחים הרבה יותר זמן בחיות שביעתיות ממה שזה לוקח בביצה.
כאילו שהעובר נראה דומה, כאילו להגיע לשם.
כן, להגיע לשם זה לוקח הרבה זמן. ההסלמות, זה שלב ההשרשה נעשית, יש השרשה, אין השרשה. תהליכים שלא קשורים בהכרח בהתפתחות העובר עצמו, אלא גם בהתפתחות מה שנקרא ממברנות החוץ-עובריות שצריכות להיות השיליה.
ודבר שני, יותר פילוסופיה, מה שאמרתי, שאנחנו לא מהנדסים שמשקרים למערכות. זה משהו מאוד כללי שצריך, האם כאילו, בגלל שזה באמת מערכת שאני פרטה את זה לאורך המון זמן, אבל כולם מתחילים דומה ומשתנים לאורך הדרך. אז האם הם מתחילים דומה בגלל שהאבולוציה לא יכולה ללכת אחורה? כאילו, האם חייבים לעבור, המערכת חייבת לעבור דרך השלבים ההתחלתיים האלה בגלל שככה היא התפתחה?
אני חושבת שהיא פשוט הגיעה לאופטימיזציה בצורה כזאת. אני לא חושבת שהיא צריכה לחזור אחורה, אני חושבת שהיא עושה את זה יפה מאוד. אם מותר לי לענות בצורה כזאת.
לא, אני שואל נגיד...
זה מוכרח לענות ככה, אבל...
לא, אני שואל נגיד לגבי הזנה, שלא מסתמכים בזנה, אבל בגלל שהיה פעם...
לא, גם הזנה נעלם אצלנו, כן?
כן, אז לא היה אפשר להגיד שמלכתחילה לא יהיה. אז אני שואל אם זה בגלל שאי אפשר לחזור אחורה ככה.
יכול להיות שבעוד מיליון שנה, הדברים של האדם לא יהיה להם.
זה כן להשתנה גם בשלבים.
בוודאי, לדוגמה, אני אתן לך דוגמה שהיא כן רלוונטית לקורס. לדוגמה, כשאתה משווה את הטרנסקריפטור, את הגנים, באיברים שונים, כן? כך כבד, כך מוח, כך בין ספישס שונים. כשאתה לוקח כבד, מסתבר שהשונות בגנום של הכבד בין ספישס שונים מאוד קטן, מאוד קטן. כשהכבד איזשהו כבר הבין את הפואנטה, הוא יודע מה הוא צריך לעשות, הוא מנקה את כל הרעלנים של הגוף, האנזימים, המים, כן, הוא בסדר. המוח, הגנום שלנו מאוד מאוד עדיין בהתפתחות. מאוד עדיין בהתפתחות. הוא עדיין לא הגיע לאופידציה, וזה לא מפתיע. זה לא מפתיע. אוקיי? אז הכל פתוח. האבולוציה לא מסתיימת. זה שאנחנו לומדים, זה לא אומר זה קצת לתת לכם קורס באבולוציה, זה כל כך זה המון handwaving, כן? אבל זה מקסים. למי שאוהב להתפתח.
יש אפשרות להעלות את המצגת?
בטח,
שיהיה לנו עכשיו לשיעור הבא שנוכל
תעריך אותה. כן, כן,
לא
כן.
על כל סלייד?
לא, אין שום בעיה. לא.
אבל למה את אומרת שיש התפתחות
רגע, אני יכולה להעתיק לך אותה כשהיא פתוחה? לא רוצה לסגור אותה?
כן.
כן, בוא ננסה, בסדר.
אבל למה את טוענת שיש עדיין התפתחות של המוח?
אני לא אומרת שיש התפתחות, יש
שעדיין,
אבולוציה.
אוקיי, אבל שעדיין תהיה התפתחות נוספת של המוח.
כי האבולוציה לא הסתיימה. אתה חושב שאנחנו השיא של השיאים? כנראה שלא. אתה רואה בין קופים לבין אדם כבר שינוי. מאוד משמעותי. נו, זה... איפה לא מצאת את זה? אני רואה שמישהו... אני קראתי לזה כדאי שתלמד אותו. בסדר, את השני אני עוד לא נותנת לך, כי לפי מה שיצא היום, אני אדבר על מה ש... אני אחליט ביום חמישי. הדבר הזה הוא מאוד דינמי, אני לא...
זה שונה ממני מה שעכשיו.
כן.
כי יש לנו מצגת של השנה שעברה.
כי מה שביקש ממני, זה, הוא אמר שהחבר'ה מאוד נהנו, אבל הייתי יותר ספציפית. אני לא בטוחה שאני מאמינה בזה, שהנוטים קצת מכל דבר. אבל מצד שני, זה חבר'ה שלי, פיזיקאים, מתמטיקאים, אולי זה מה שצריך לעשות. אני עוד לא יודעת, אתה יודע, זה נושא בהתהוות. אתם תגידו לי. כן? כי להיכנס למשהו מאוד מאוד ספציפי ולהתחיל לתת לכם מולקולות, הנה, שנה שעברה דיברתי על פתולוגיות, שתבינו גם שיש משמעות להתפתחות, גם למומים מולדים, שזה משהו מאוד מאוד רלוונטי. השנה אני לא אצלי. אני גם, אחרת אני משעמם את עצמי, אז אני כל שנה מחליטה.
על ההיווצרות של המולקולה הזאת. זה יותר הגיוני,
מן הסתם,
מכניזם.
זה עצם ההתפתחות של אותו מבנה שנקרא הנוטוקורד שבגינו. עכשיו, הנוטוקורד בעצמו זה משהו שהוא מאוד 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 ישן, והוא אורגנייזר של כלל הגוף שלנו. אז למה סוניק דווקא מתבטא באותו אזור שנקרא הנוטוקורד? באמת אף אחד לא יודע, אבל עובדה היא שיש מרכזים בגוף העובר שנקראים אורגנייזינג סנטר, ואותם אורגנייזינג סנטר קיימים מורפוגנים, אוקיי? וסוניק הוא אחד מהם, כמו שאמרתי, DNP, ווינטק, GF ואחרים, ואלו אחראים ליצירת דיגום שלם של הגוף. במקרה של הנוטוקורד, מערכת העצבים. גם סוניק, דרך אגב, עושה לבלב. הוא פועל על האנדודרם והוא יודע ליצור לבלבים. זאת אומרת, הוא עושה המון דברים. הוא יוצר ורטברה גם כן, הוא כמו שמש. הנוטוקורד זה כמו איזה מקל כזה, שממנו יוצאים מולקולות לכל הכיוונים. ולאן שזה משפיע, שם הוא עושה אורגניזציה. מעבר לזה?
אי אפשר לעשות את זה על סביבה מלאכותית?
לא. לא, ניסו. לא. זה מעניין, זה היופי. זה היופי.
לא רוצה להגיש את ניצב קומר מפילדלפיה. אין לו פרסום. אין לו פרסום.
יש עדיין נמצא הוא מתקדם בכלל לא רע. אני חושבת שלשנה הבאה.
הנה שבסוף נובמבר יש לנו דדליין.
לא, אני לא אצא לזה השנה, בעזרת השם מה שנקרא, שנה הבאה להתקדם יפה.
אז מאי.
הוא כבר קולט, למאי, תראי מה שאתם תוציאו את הזה, כל עוד יש לו את הרצה, כן, אז אני מגישה לו את הרצה.
זה שני מועדים קבועים בשנה, סוף נובמבר ותחילת מאי.
בסדר, טוב, אתם תודיעו לנו מראש או משהו, אני אשמח לעשות את זה. על הכיפאק, תודה. תודה, אקו. בהתחלה אמרתי לו, מה הוא מדבר? ואז
יש דרך בסיסאית לגלות כאילו להראות לו? אני פשוט עשיתי רע,
Okay, I realized that I had the microphone and I didn't use it, so now it may be better and I won't. Okay, so the process that we dealt with in the first hour was basically this process of induction, neural induction, and fate determination. Now, the third process that I would like to deal with, and I don't know whether we are going to get to all of them, is basically cell proliferation. So as I said, all these progenitors are not yet differentiated, and one of the next stages in the development of the spinal cord and brain would be the actual growth of the cells, the actual division of the cells, and this occurs via a very, very precise process that characterizes epithelia. I may say that in this sense, the nervous system is not peculiar because it happens in all kinds of epithelia, or what we call pseudostratified epithelia. I'm going to explain you this in a second. So two examples of pseudostratified epithelia, you don't know what this is, I'll clarify it in a minute, are, for example, the neural tube, which we are dealing with. This is, by the way, how the notochord looks like. It's not an epithelium. And this is a structure on both sides of the tube, which is called the somite. We work on both of them a lot in the lab. The somite is going to generate all the muscles of your body, the vertebra, the ribs, many blood vessels, endothelium, smooth muscle, and so on and so forth, the dermis of the back, etc. And both the neural tube as well as the early somite are composed of what we call pseudostratified epithelium. I'm going to explain it. Because the neural tube basically is generated not only, again, a sheker lavan katan, is formed by invagination and closure. By definition, it leaves a little hole, which is going to become the central canal of the spinal cord, if you know what this is, or the ventricle in your brain. Okay? You have ventricles
lateral ventricles, third, fourth ventricle. This is generated by the actual folding of the neural tube. And you have it here. This is the ventricle, which is in the embryo quite virtual, okay? Because the cells grow and then they obliterate the ventricle and they leave only a very tiny hole. But this is the ventricular part, which is also called apical. This is an important issue, apical. While this outer part of the epithelium is called basal. So in a pseudostratified epithelium, each single cell spans the entire apicobasal domain of the epithelium. So all this is one cell. Now, why do we call it pseudostratified epithelium? Why it is pseudo? Because the cells look like this. And I'm going to draw it wherever I have left this little space here. Because if this is a cross-section, and this is apical, and this is basal, each cell will go like this. But, for example, in this cell, the nucleus is going to be here, very close to the apical part. In its neighbor, the nucleus will be here. In the next neighbor, the nucleus will be here. So it looks like pseudostratified because when I look at it and I have many, many, many cells, it looks as if I had 1, 2, 3 layers of cells. But it's not like that. A stratified epithelium has many cell layers. So it's 1, 2, and 3. But here it is pseudostratified because each cell, okay, spans the entire apicobasal domain. So it's a one-layer epithelium. But the nuclei are interspersed at different apicobasal areas. Why is this like that? Why? We asked why. I have an answer. I have an answer for a why. Okay, so this is the way it looks like. And this is what happens. It appears that this is very beautiful concerning cell proliferation. Did you ever study the cell cycle? Phases of the cell cycle? Okay, so for those who know, I'm going to try to be very basic, okay? If this is the apical side and this is the basal side, inner and outer, the cells undergo mitosis, actual body cell division, in the apical side. Let us begin by here, okay? So mitosis will take place in the apical side. And the nucleus will obviously need to go to the apical side for the cell body to divide. But then the nucleus begins moving towards the basal side of the epithelium while it undergoes a shift in the cell cycle properties. Namely, it goes from mitosis. Now I have to go back here. Let's say mitosis has occurred here. Then the cell will go into a gap phase, which is called G1, in which the cell prepares itself for a round of DNA replication. Because the cell will be 2N chromosomes, will have to replicate itself to a 4N chromosomes, and then again to 2N chromosomes when the bodies divide. So this is a gap phase, and this, when the cell is located at its utmost basal location, it replicates its DNA. And this is called the S phase of the cell cycle, or the synthetic phase of the cell cycle. Then it prepares to mitosis by going into the GAP2 or G2 phase, and then it goes to mitosis. So the pseudostratified epithelial appearance occurs because of this movement of nuclei from the apical to the basal and back to the apical side of the epithelium during the different phases of the cell cycle. This nuclear movement has a name. It is called interkinetic nuclear migration. Interkinetic nuclear migration, INM. It occurs only in pseudostratified epithelia. And for you to remember is that these epithelia are composed of one single cell. Okay? So, what characterizes an epithelium and how do we proliferate? We can proliferate in two ways. And look now at this part here. We can proliferate in a symmetric way, and I'm talking about this type of epithelium, in which the cleavage plane, or basically the furrow of the mitosis, will take place in a vertical manner, so as the two cells that divide or that are generated from the event of mitosis remain apically located. In most cases, as beautifully shown by Sue McConnell years ago for the cortex, in most cases, in the early development of the cortex, most of the proliferations are going to be symmetric. Most of the cells that result from the event of mitosis will remain in the apical side, and these are symmetric, namely one progenitor will generate two progenitors, which are equal to its mother cell. And this will account to actual growth of the structure without differentiation. Namely, we are talking about the first stage in which we need to increase the number of cells without taking yet any fundamental decisions. So this is symmetric divisions. And this will, as I said, create two cells which are likely to be equal. Now, this model has been contested, and we know now that in different places of the brain, even if the division is vertical, let us call it vertical, it is not symmetric necessarily. Symmetric means fate, and vertical or horizontal means the plane of division. So you can still divide vertically. It doesn't mean necessarily that you are going to generate two identical daughter cells. So this is for you important. Also, we see that in cortical development, in ferrets, this has been done in ferrets, most of the initial proliferation events are vertical, and this is indeed related with symmetrical outcomes. In the late development of the cortex, there are still symmetrical divisions. There are many more horizontal divisions as well, and these symmetrical ones do not necessarily generate equal progeny. The other type of division you may have is asymmetric. Namely, first of all, you have, let me take it back. The other type of division is horizontal, the horizontal plane, which will generate two cells, one keeping contact with the apical domain and one going basally. Now, in the cortex, again, if we go to the same example, we know that in the late cortical development, this cell will remain as a kind of neural stem cell, which will generate further generations of divisions, whereas this one, the one which is far from the apical domain, will differentiate and go to the basal side where neurons are produced. Okay? Now, I want you to keep in mind what I said before. Not every vertical division will be symmetric. Not every horizontal division will be asymmetric. But during development, there is a clear shift between vertical and horizontal, which are in fact symmetric and asymmetric. So statistically speaking, this is most of them, certainly not all of them. So this is the idea. Why a type of vertical or horizontal division will be asymmetric? Can anybody of you give me an idea? Can you think of a mechanism that will turn one dividing cell into two unequal cells? Can you think about a general mechanism? Okay. Gravity?
A cell that looks like this, and at the bottom you'll have fewer more cells, and at the top a few or more cells, more material, and at the top a little bit less. So you get a sort of tinier and a little bit bigger cell.
Okay. Okay, a gradient of concentrations, gravity.
Some kind of outside
an extrinsic signal? Additional ideas? All of them are okay, right? So we'll elaborate a little bit. Okay, let's begin by gravity. I wouldn't call it gravity. We have no evidence, at least, of any gravitational thing because the embryo moves all the time. So gravity would mean that something is static and there is always a gravitational field. So it's not gravity, but I think you made your point because it's contact with the apical domain. Okay, contact or lack of contact with the apical domain. We know that in the apical domain, that cells which are pseudostratified are very different, and we work a lot on that in the lab. This domain of a cell is very different from this domain. Just to illustrate my point, for example, the apical domain of a cell, of a pseudostratified cell, contains molecules like N-cadherin, which are adherence junction molecules, those that keep the cells together. There are tight junctions, molecules like ZO1, zona occludens 1, molecules like N-cadherin, APKC, and others, whereas the basal part of this cell will express matrix molecules, basement membrane molecules, such as collagen 4, laminin, and so on and so forth. So we are talking about a cell which is very different on the two poles. So if you divide the cell in this way, okay, this part of the cell, the basal daughter cell, will inherit different determinants than the apical domain of the cell. And the question is whether during the process of regenerating a pseudostratified epithelial cell, will it regenerate everything back? Yes. If it generates this, it will regenerate everything again. But if it stays long enough in this area, it will not regenerate. And if it doesn't regenerate, it will go to differentiation. Growth factors. We know absolutely that there are growth factors, and we'll talk about one of them, molecules that call the cells to migrate out at a certain point in development, that they are substrate like radial glial cells on which cells are going to migrate in order to arrive to the different basal areas of the epithelium, okay? So that many factors which are extrinsic to the cells are able to change the orientation of a division of the cell, okay? And this will appear later on and not at the beginning. Okay, something else has been said, different molecules that are expressed in a gradient. I think I have touched upon that. Cells, molecules that are here, others that are there. And let me tell you that some of them are also transported. There is a molecule also coming from Drosophila existing in human beings, which is called LGN. LGN is a molecule which is first expressed in a prophase cell. In a certain stage of the cell cycle, it is expressed everywhere in the cortex of the cell. But then when the cell is going on to mitosis, it is going to the apical domain because another molecule called inscutable brings her by hand to the apical domain. And depending on which cell will inherit LGN, okay, the cell will become either two progenitors or a progenitor and a differentiating cell. So again here, without entering into details, there are many, many mechanisms that can affect the differentiation of a cell, and look at this. Okay? What you see here is how a certain determinant, which is the blue, the white molecule, during a process of mitosis, will segregate asymmetrically between the two daughter cells. And this means that the upper cell will now become different from the lower cell, because there has been an asymmetric segregation of a determinant. Cadherin is one of them, etc., etc. So again here, this is an introductory talk. I gave you just the concept and I gave you just the idea without entering into the details. But let me just say that orientation of cell divisions plays a role. The asymmetric allocation of different intrinsic determinants, the activity of extrinsic growth factors on influencing the plane of orientation of divisions, and the transport, the intracellular transport of different apical and basal determinants, which basically will determine the asymmetric versus symmetric outcomes of dividing cells. The cell has divided. One of them is going now to become a neuron. So how does it migrate? Because all the cells, as I said, are produced in the apical domain, but the cortex or any tissue in our brain or spinal cord has to grow. Now, in the brain, the growth is basically from inside to outside. Namely, the cells which are localized in the basal part of the brain are the youngest, and the ones that are localized inside are the oldest. So the cells need a kind of substrate onto which they have to migrate and move in order to cross all the layers of progenitors and to get localized superficially. And this is given in the brain and spinal cord by a kind of cell called the radial glial cell, which is a cell which, again, spans the entire extent of the epithelium. It is one of the cells that is born first in the developing brain. And on top of this cell, the progenitors of the neurons begin basically migrating to generate the different layers. And this is one example. This is cell migration in the cortex. So you can see the ventricular zone. This is apical, and this is basal. Okay, this is out, and this is in. So here you have the different layers of the cortex forming. You know that we have 6 layers in the cortex. And the cells which are the oldest localized in, and those which are youngest migrate out into the cortical plate. Now, a molecule that is very important for this inside-out migration is called reelin. Reelin is a molecule which induces a set of metabolic and biochemical events, which, again, it's not the place and time to discuss. But if we produce a mutant of reelin, which is called the Riller mouse, okay, what we get is a completely distorted formation of cortical layers, obviously with behavioral and very important cellular and tissular consequences. Why? Because the normal migration of the cells towards the basal domain of the nascent cortical plate has not taken place. There are many other molecules which we know participate in migration of neuronal progenitors, which participate in migration. Again, I'm not going to talk about. And we have many mouse mutants in which migration is completely distorted. I'll give you one example. There is one molecule. I'm not going to give you the details, which is responsible for migration of cells and which is responsible for the formation of the sulci and gyri of the cortex. You know that our cortex is not smooth. I mean, in the embryo, it is initially smooth. But as the number of neurons begins increasing, we have to accommodate within a given volume, which is your cranium, you have to accommodate many, many neurons. How do you do it? By creating involutions. And by this, in this way, you accommodate a maximum of neurons in a given volume. So we have sulci and gyri. And this is given by this differential migration of cells and by cell proliferation. And there is a mutant or a disease in children which is called lissencephaly. Lissencephaly is called smooth brain, in which children are born with smooth brains. And this is not due necessarily to a process or to an initial defective proliferation of cells. It is due to defective migration of cells and defective formation of sulci and gyri. Not going to talk about the molecules. We know the molecules. We have mouse models that account for this. And this is to illustrate how migration of cells is important for the proper formation of the brain. You see, Mickey, this is what you do. You talk about a process which is so crucial, and you talk about it for 5 minutes. This is frustrating. Let's go to the next topic in the frame of this. כן, בבקשה. No, go on.
Slide before, so there is no involution to humans that just burn the
Oh, no, 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 no. No, this is not this slide, by the way. This is the Riller mutant. Riller mutant has nothing to do with sulci and gyri. I'm talking about the lissencephaly mutant without mentioning molecules. No, your brain is smooth, I would guess, till mid-gestation. And then it begins getting sulci and gyri. Until when? Until after birth. Until after birth, because there are structures which the brain doesn't stop being formed in the embryo, okay, in the fetus. There are structures, for example, your cerebellum, okay, which is going to grow a lot after birth as well. So most of these beautiful, fancy forms of your brain are formed from mid-gestation onwards and are going to continue. Okay, and as you know, there is neurogenesis even in our brains. Also, it seems to me quite. I mean, I don't feel I'm more clever every day, but you know. Okay, but this is again another topic. Another important topic is axon guidance. The neuron has formed. The neuron has reached its definitive location, and now it is going to create basically synapses. But before it creates synapses and before it begins functioning, it has to grow a neurite, a dendrite, axons. And these axons have to know where to go because they have to form circuits which are coherent and are very accurate. And this field, which is a field of research embodying a large amount of researchers, is called axonal guidance and axonal connectivity. So what is an axon? It's a neurite. It's a protrusion from the cell body. It can be an axon. It can be a dendrite. We call it in initial development a neurite because initially you don't know always if it is going to develop into an axon or a dendrite. And this is the way it looks. This is the tip. The tip of an axon is a very curious area. It looks like a mesenchymal cell which looks for food and looks for appropriate places to grow. So it has philopodia. It puts up all kinds of prolongations which basically sense the environment. And if the environment says, come, 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 this prolongation will dominate and the entire neurite will go in that direction. But if the environment says, don't come, don't enter, this prolongation is going to retract and the axon is going to go elsewhere. So look at it as a very curious personality that looks for a good environment but basically retracts from environments which are hostile. These environments are molecules. And these molecules act via receptors. And we know now about more than 10 pairs of receptor molecule pairs, Slitten, Robo, Netrin, and DCC, Sonic and its receptors, which also play a role in axonal guidance, etc., etc. I'm not going to give you more than that, which are known to guide axonal growth at different stages. And I'm going to give you one example, which is beautiful, for which we know a lot. And these are interneurons. So again, we go to our spinal cord, okay? And there are interneurons, which are called dorsal interneurons. They receive the information from sensory ganglia, from the periphery, and they relay this information to the brain. For example, I am burned, and I say I. Okay? Which type of neuron enables me to say I, it hurts? These are interneurons, which are located here. These are dorsal interneurons. There are different types, 6 different types. We are looking at one of them. Okay? It doesn't matter. So this neuron has a very funny behavior in terms of axonal growth. This is the cell body. The cell body first begins to send its neurite down ventrally as if it would escape the dorsal portion of the spinal cord, and then goes down, and all of a sudden it turns towards the floor plate. This is the floor plate. This is the central canal. all of a sudden it turns towards the floor plate, and then it decussates. Namely, it changes its direction from the ipsilateral side of the spinal cord to the contralateral part of the spinal cord. Namely, it goes to the other side of the central nervous system, and then it goes to the brain. This is very stereotypic. It occurs all the time for the same type of neurons. You can see it here. If we mark these dorsal interneurons, you see the bundle. This is not a neuron. This is a bundle of dorsal interneurons marked by a certain reporter that marks them, a certain molecular reporter. You see them going here, going there, and crossing. And if you open the neural tube into a preparation that we call the open book, okay, it's like opening the spinal cord into a flat type of movement. What you see is this crossing the floor plate. This is in gray, the floor plate, and the fibers go towards the brain. This is what you see here. You see these fibers growing towards the brain. So how can we explain, and how many molecules do we need to make this complex and accurate stereotypic type of behavior? So let me explain you what we know. This took like 20 years of research, guys. Okay, and again, all these molecules are expressed in Drosophila. Again, if I may point to the evolutionary idea or concept. So let's go one by one. I'm not going to detail on everyone, just give you the idea of complexity, but complexity with accuracy. So I told you before that BMP is expressed in the dorsal part of the spinal cord, okay? And it is an antagonist to sonic hedgehog. They fight all the time at very early stages to determine different fates. And BMP at the beginning will determine dorsal fates. It will be part of the determination of dorsal interneurons. But once these neurons are determined, BMP and sonic continue to be there. What do? They don't disappear. They do different functions. So one molecule, many functions, based on the time we are dealing with. This is another concept that you should remember. And this is why so little amount of molecules have so many different effects depending on the timeline. So BMP, what it does is it repels the axons of these dorsal interneurons once they are fully differentiated and they begin growing an axon. So the first molecule playing a role, which is a repellent role from the dorsal area of the spinal cord, is BMP. So if you put a lot of BMP, you're going to repel all the axons away from the midline, and they are going to grow ventrally. Now, there is another molecule. Somebody can elaborate here? Why does the axon grow this way and makes this turn? Perfect. So it appears that there is a molecule called netrin-1. There are a few netrins, but netrin-1 is the responsible, which is expressed in the floor plate. So the floor plate not only expresses sonic, it expresses netrin. So netrin-1, sonic plays also a role. We are not going to deal with it now in the context of commissurals. So netrin attracts, it's a secreted molecule, it attracts these fibers to grow towards the floor plate. And the receptor of netrin is called DCC. DCC is also a molecule in colon carcinomas. This is how the name comes. I'm not going to develop any further on that. It's quite complicated. There are different receptors for this molecule, but it attracts the growth cone to come towards the floor plate. So we have already a mechanism of repulsion. We have also a mechanism of attraction. Interestingly, DCC, the receptor, is expressed not immediately here, but once the growth cone begins approaching the more ventral areas, indicating that low amounts of netrin already induce the expression of the receptor. So the fiber arrives and reaches this area. Okay? At this point, let me make the long story short, this fiber is going to cross the, yes.
You want to say?
No, Kadima.
Sure. If there are no DC receptors yet, how can then a gene cause the formation of the receptors if it doesn't have any receptors to bind?
Okay, the genes for everything exist in the cell, right? You know that each cell has the full repertoire of all the genes, but the gene is somehow silent. In many events in development, the existence of the ligand upregulates the gene, because otherwise each cell would have to produce all the time all the 25,000 genes, which it doesn't. But this is another topic. Gene transcription and regulation of gene transcription is a very complicated thing. This is a question in itself. When and how does a certain receptor begin to be expressed? So one mechanism is that the ligand itself begins upregulating the levels of the receptor, right? Okay, so the mechanism that enables this cell basically to cross is again a repellent mechanism. At a certain point, this cell begins again expressing a molecule which is called ROBO. ROBO, or roundabout, in Drosophila is a receptor, and slit is expressed in the floor plate. So when the cell begins expressing ROBO, it crosses the midline and it is repelled from this area. Repelled from this area. And then it goes to the other side. And then it goes to this side and to the brain. How does it do it? It does it by virtue of a gradient that exists along the longitudinal axis between two molecules. One is the famous sonic hedgehog, and the other one is wind. Because sonic hedgehog at this point in development is expressed in the floor plate in a graded manner, and wind is expressed in a opposite gradient from head to tail. So we believe that these fibers are somehow attracted by an increasing concentration gradient of wind, thereby arriving or reaching the brain. And I'm not going to develop any further because FF rings are additional cell contact mediated molecules which contribute to fasciculation. Namely, the moment additional fibers have joined the club, they have to grow as a fascicle, as a bundle of fibers. And FF rings play a very important role in that. So look at a process such as axonal growth. Think that the millions of fibers that grow out from neurons, from nascent neurons, have to make the proper choice and have to reach the proper areas. How do we do it? By virtue of many very highly regulated, tightly regulated processes. Let me skip this. Okay, let me deal with this for a moment. And this is neurons versus glia. Neurons versus glia. In most of the nervous systems, neurogenesis precedes gliogenesis. But as you know, we have many types of glia in the brain, which are extremely important and play very important functions. Let me again go to the previous slide if I've decided to deal with this. And this is the following. Glia is divided into oligodendrocytes, into astrocytes for the brain, and they all develop in the central nervous system. Oligodendrocytes are those which originate the myelin in the brain, and in the peripheral nervous system that we are going to learn next week, there are cells which are called Schwann cells, and these cells are the ones which are responsible for myelinating peripheral axons. So the process of myelination is different in the CNS and PNS, and different cell types with different origins are responsible for that. So these are CNS cells, and the Schwann cells arise from embryonic progenitors that we investigate quite a lot in the laboratory that are called neural crest cells, about which I'm going to try not to speak today and just to refer to during the next week. So if we talk about gliogenesis, let us look just at one slide. It's very complicated. I'm going to deal only with one aspect. At the beginning, we have a kind of neural stem cell. Again, what I would call a progenitor. בסדר? So this progenitor cell is going to generate, the stem cell is going to generate a neuronal progenitor, and this is going to generate a neuron. And as I said, in true life, neurogenesis precedes gliogenesis. Why does it precede, or how? This why bothers me now after this question. How does it precede? It's because gliogenesis is basically inhibited. In development, many, if not most, of the crucial processes occur via inhibition and not via stimulation. Sometimes, in order to respect the timing of development, you have to be inhibited all the time. And it is only when you are released from inhibition that something takes place. And this is because when you become a neural progenitor, you get neurogenic signals. And these signals are called proneural genes. For example, proneural genes, neurogenin, MASH1 or CASH1 for the chick embryo. There are a series of genes which are transcription factors, and they will promote neurogenesis. But at the same time, sonic promotes a certain class of factors and inhibits another. These proneural genes will promote the production of downstream neural genes, but will inhibit the production of others. It doesn't work. I'm realizing that at the end of the talk. So gliogenesis, which is the alternative pathway, is not taking place because it is inhibited. But then something happens. Neurons are formed. And then there is a magic process which is called lateral inhibition. And this, let me give you the example of a ganglion, which is a peripheral pool of neurons, which derives from the neural crest. This is a sensory ganglion in which this has been thoroughly demonstrated. This is a ganglion, and we have a certain amount of neurons, and these are already formed. Now, the progenitors, which will become glia, by the way, localize initially in the periphery of the ganglion, because the ganglia are formed inside out. Now, once these neurons have formed, and there is a tight relationship between the neurons and these progenitors, the blue progenitors, there is a process that is called lateral inhibition. Namely, by touching one each other, there is a process. Let me draw now two cells in high magnification. There is a process by which a molecule called delta activates a receptor, which is a transmembrane receptor called Notch. Now, if delta is high in a stochastic manner, and in every neuron this is the case, if delta is high in the neuron, it will upregulate Notch, which is its receptor, in the neighboring cell by touching it. It's a cell adhesion-mediated process. And by saying that, delta will say the other cell that upregulates Notch, do not become a neuron. So in the cell in which Notch is high, delta is going to be low. And in the cell in which delta is high, Notch is going to be low. So this is a bidirectional lateral inhibition mediated by a very famous signaling pathway called the delta Notch pathway, again already operative in Drosophila, which will decide that this cell is a neuron and this one will not become a neuron. And if it doesn't become a neuron, it will become a glial cell. So this is not the only story, because this cell which will become glia will also receive additional glial growth factors that will enable it to differentiate fully into glia. I'm not going to talk about them. But the initial step is don't become a neuron. And the alternative pathway will be a positive one, which will be given also by the neuron, and I'm not going to mention the name of the molecule, okay? So, lateral inhibition will inhibit the proneural genes and will relieve the inhibition on gliogenic signals. So, inhibition of inhibition is excitation, if you wish, in neurobiological terms, and then gliogenic signals will become available to the cell, and they will lead to the development of glia only after neurogenesis has taken place. I'm not going to deal with the stem cells, which is a completely new field. Let me just introduce you to the neural crest in the dorsal neural tube. This is the tube, this is the lumen. There is a population of cells residing very early in the dorsal top of the neural tube, which will not stay in the central nervous system. They will undergo a process of epithelial to mesenchymal transition that we have studied in the lab and that we have discovered its transcriptional machinery. And these cells are going to leave the dorsal neural tube, and they are going to migrate extensively throughout the embryo and generate an extraordinary variety of derivatives. Among them, if this is the neural tube, they are going to generate sensory ganglia, both dorsal root ganglia along the trunk and all the cranial sensory ganglia. They are going to develop into sympathetic and parasympathetic neurons and glia. They are going to generate the chromatin cells of the adrenal medulla, which are responsible for the fight and flight response. They are going to generate the entire innervation of your gut and lungs. They are going, obviously, to monitor peristalsis of your intestines, the rate at which your heart beats, the pressure, and many, many other things. And something which is very, very neat, they are going to generate the melanocytes of your body. All the pigment of the body, the beautiful color of birds and of fishes, all this beauty is neural crest. And it is very interesting how a cell which is neurally minded is going to generate pigment. And we have studied this very in depth in the lab, and we may touch upon this in the next course that we have. And additional derivatives that I'm not going to deal with. And look at how these cells emigrate from the tube. They emigrate in a segmental manner. This is the hindbrain. This is what is going to develop into your cerebellum. And this is how the cells will emigrate and will leave the dorsal neural primordium. I'm going to finish with this. I'm not going to enter higher brain functions that I meant to. Let me just finish with cell death. Okay, I cannot leave you without the idea that cell death is not a bad thing. In development, it's a major process, and it is of utmost importance for developing a normal nervous system. Now, cell death occurs normally as a result of competition for factors that lie or are produced in the periphery. So a cell, a neuron, will send its axon. The axon will reach the periphery, let's say the skin, if it's a sensory axon. And then many, many axons will arrive, more than needed. And these axons will ask the periphery, how much food can you give me? And it appears that the food that the periphery gives you is limited. This is the idea which is called the neurotrophic hypothesis. The amount of food is limited. So if the amount of food is limited, we are going to fight for this food. And if we are 10 axons, only 5 of us is going to survive and to innervate, while the other 5 are going to retract. And if they retract, they are going to send to the cell body sitting in a ganglion or in a nucleus in the CNS a message, we lost the battle. And if we lost the battle, we are going to die. Why is this important? This is important because the size of a ganglion or the size of a neuronal pool in the CNS has to match the size of its target. Whether it's because we have to act in a harmonious way. Now, why does the system create more neurons than necessary? We don't know. We don't know. Maybe this is also an evolutionary idea that we have not yet reached any type of optimization regarding that, but this is what happens. Okay, so we create at the beginning more neurons than necessary. We send more axons than necessary. We even make more synapses than necessary in some events. And then we have retraction by neurotrophic factors, which are limited, and also synapse elimination. At the end, remaining only with those in which the activity is the highest, right? And this is called synapse elimination. But let us see this, and with this we are going to finish. So this is an embryo, okay? If we remove the leg, not the leg, the limbat, what is going to become the leg of this embryo, okay? If at the beginning we generate the same amount of motor neurons here, if we remove the leg, only 10% is going to survive, and in opposite the missing limb, we are going to have very few motor neurons left. So less target in the periphery, less motor neurons left in the spinal cord. On the contrary, if we put here an extra limb, and we can do it very easily, okay, we graft here an extra limb, what we see is that if normal survival is 50% in the untouched side, here we are going to have like 70%, 75% survival, because the target is bigger than normal. And these are experiments, classical experiments, and you should read this book, classical experiments made by Victor Hamburger in St. Louis University and followed by Rita Levi-Montalcini, Nobel Prize, by virtue of her discovery of nerve growth factor, one of the neurotrophic factors that is responsible for that. NGF not for the motor neuron survival, but for sensory survival and sympathetic neuron survival. Okay, and the guy, Steve Cohen, who with her found and biochemically purified NGF, which was the first. Now we know of many neurotrophic factors, BDNF, NT3, NT4, NT56, many growth factors that are able to do the job, and the neurotrophic hypothesis still holds till now. It has been refined. Again, this is a matter for an entire course. Another thing which is important is activity, neuronal activity. We know that in order to make a synapse and to maintain it, we need the fibers to send impulses, to send action potential, and this is what activates and maintains the synapse. If we paralyze the embryo, we get 75% survival, less survival. So activity at a certain point will, in addition to neurotrophic factors, which go all the way from the periphery retrogradely to the cell soma, will eventually account for the survival of those cells. Now, this is called apoptosis. It's a very particular type of death, and it is programmed cell death. Why? Because it takes place stereotypically at a given time and at different extents in different places. For example, the ganglia and the motor neuron pool in this area are smaller, whereas ganglia, peripheral ganglia and motor neural pools of these areas innervating the limb are larger. Normal cell death is lower here than here. Okay, so we know that also based on normal developmental features. Regulation of cell numbers, synaptogenesis, synapse